Okay, now apparently it worked out. It's truly a pleasure to have here Yichao Zeng for our last talk in our seminar series of the graduate program in ecology from USP, Universidade de São Paulo. Yichao is originally, he did the undergrad studies in Peking and then moved to US, did a master's in University of Florida, and now is currently a PhD candidate at the University of Arizona. He has a very interesting publication record from using deep learning to understand, to identify individuals of giant pandas, to try to connect traditional medicinal herbs with conservation for endangered species, to macro evolutionary dynamics of ecological interactions. Yichao is visiting my lab. It's a virtual visit, unfortunately, but we have been able to interact a bit. So it's a very interesting PhD research. He's now developing, and he will talk a bit about that today for us. Thank you so much, Yichao, for your willingness to give a talk to us. And well, the room is yours. You can start whenever you're ready.
Thank you very much, Mildu, for the introduction. Today, I'm very happy to talk about my research on testing hypotheses that link macroevolution and species interactions. Okay, so what is macroevolution? The term macroevolution was invented simultaneously with microevolution in 1927. Since the mid-20th century, it has been widely used to refer to evolution above the species level. Macroevolution generally occurs over geological timescales, that is, over millions of years. And what are species interactions? Species interactions are also termed interspecific interactions. They are interactions between different species within biological communities. Conventionally, all possible interactions between species can be put into one of the classes shown on the right, based on the interaction's net effect on participants. These classes include mutualism, predation, parasitism, herbivory, competition, commensalism, amensalism, and neutralism. It is not uncommon for an interaction between a pair of species to belong to multiple classes. This is because outcomes of species interactions on the participants are often context dependent in both magnitude and direction. For example, the interaction between ants and aphids is often described as a mutualism in which ants protect the aphids from enemies, while the aphids provide honeydew for the ants as a sugar reward. However, this interaction is not always beneficial for aphids. In the absence of enemies, this interaction can be harmful for the aphids as well, even though the mechanism of interaction between aphids and ants remain the same. There are also many cases of mutualisms grading into parasitism. For example, cheating in mutualisms. So there has been a long interest in understanding how species interactions influence evolution. Hembury and Weber 2020 provides a very great review on this topic, which I find very inspiring. So here I'm going to give a very brief overview of the historical roots of such investigations. And this part of my talk is largely based on this 2020 review paper, but it contains some of my understanding as well. So in The Origin of Species, Darwin used examples of competitors and enemies to explain how organisms are in a constant struggle for existence and are subject to natural selection. Before the 1960s, some important authors involved in the modern synthesis, such as Rinsch and Simpson, had recognized the macroevolutionary significance of species interactions. During the 1960s, there was an increased interest in this topic, and some of the most important hypotheses were proposed during this period. Such hypotheses include the famous escape and radiate hypothesis by Ehrlich and Raven. They postulated that escape from plant defense gives butterflies an adaptive zone for rapid diversification, and that escape from herbivory gives plants an adaptive zone for rapid diversification. This offers a possible explanation for the exceptional diversity of herbivorous insects and plants. One of the academic traditions for the study of macroevolution is rooted in paleontology, using fossil data to understand macroevolution. This type of research blossomed in the 1970s and the 1980s, and many paleontological studies during this period looked at how species interactions affected clade dynamics. For example, there are two groups of invertebrates, one called brachiopods, the other called bivalves, who have very similar ecological niches. And what is interesting is that over geological timescales, brachiopod diversity declined while bivalve diversity increased. And this observation leads to the question of whether bivalves outcompeted about brachiopods. These authors, Gould and Calloway, argued that there was no evidence for the role of competition in this replacement, but there has been a long-standing debate over this question until at least very recently. Another tradition for the study of macroevolution is rooted in neontology, which is the study of living organisms. This tradition uses contemporary species trait data and molecular phylogenies to model macroevolution. This type of research blossomed in recent decades. Many such studies looked at traits that have important functions in species interactions. A typical study is Weber and Agrawal 2014, in which they compared diversification rates of plants with extrafloral nectaries to those without. They found that plants with extrafloral nectaries, so here's a little background of plants with extrafloral nectaries form mutualistic interactions with ants. And in this relationship, plants attract ants with sugary nectar, and ants protect the plants from herbivores in return. These authors found that plants that form such mutualistic interactions have higher diversification rates than those that do not. Another tradition, so here's my research. So for my research, I focused on 4 sources of variation in species interactions and their relationships to macroevolution. These 4 sources of variation are presence, absence, ecological nature, phylogenetic distance of partner shift, and rates of partner shift. And I will talk about them in turn. The first question I try to answer is whether species interactions have predictable impacts on species diversification, that is, speciation minus extinction. The earliest paper I could find that asks a similar question is the one published in 2008 by Jablonski. This review of both paleontological and neontological studies shows prevalent mismatches between organismic level fitness effect of species interactions and species interactions effects on clade diversification. For example, you can see here in the table on the right, this category of species interactions, predation or herbivory is positive for one participant individual and negative for the other by definition. However, when you look at their effects on the participants at the clade level, you can see that this class of interactions can have positive effects for on both participants. And this raises the question of whether and when species interactions translate into congruent effect at the clade level. So to answer this question, I used the Web of Science to search for studies that looked at the effects of species interactions on diversification. We found over 4,000 studies in this search. Then we used 3 criteria to narrow down the pool of studies. The first is that the study needs to perform some kind of statistical test for the effects of species interactions on diversification. Sister clade comparisons are also acceptable because they are essentially comparisons of diversification rates as well. The second criterion is that there needs to be ecological evidence for species interactions. Based on this criterion, we excluded paleontological studies because they usually have relatively limited ecological evidence. The third is that there needs to be variation in the presence and absence of species interactions among species. Also, although almost no macroevolutionary studies we found provided direct measurements of interaction intensity, some provided ecological evidence so that we could infer variation in the intensity of species interactions. These studies were included as well. So we excluded some studies, such as those that compared different versions of interactions that were of the same sign to the focal taxa. For example, studies that compared diversification rates of mutualistic herbivores versus antagonistic herbivores were not included. This is because even when one version is associated with a higher diversification rate than the other, it is impossible to tell whether the first version of interaction increased diversification rates or it is the second version that decreased diversification rates. And with the 29 studies we found, I did a two-tailed Fisher's exact test to see the correlation between these two directions. The first being the direction of the interaction for the taxon at the individual level, the other being the direction of the effect of the interaction on the other being the direction of the effect of the interaction on the diversification of the clade, which is the effect at the clade level. And the results of the Fisher's exact test show that there is a clear correlation between the two directions. And we also calculated effect size as the binary log response ratio of diversification rates. And here, R is the ratio of the two diversification rates. And specifically, it is the rate of the rate. It is ratio of the rate with the interaction present to the rate with the interaction being absent, or or it's stronger versus weaker. So overall, we found that some subgroups have very small sample sizes, so we are not super confident about results for those groups, but overall, we can see in the results that the effect sizes for positive interactions are generally positive, and the effect sizes for negative interactions are generally negative. So overall, we found that the presence of a positive interaction generally increases rates of diversification, while the presence of a negative interaction generally decreases rates of diversification. The second question I ask is whether the positive-negative nature of species interactions predict their persistence over macroevolutionary timescales. This is largely motivated by the long-standing debate about the macroevolutionary persistence of mutualisms, especially in comparison to that of antagonisms. I am interested to see what patterns will emerge if all known mutualisms and antagonisms are pooled and their ages, especially the oldest ages, are compared. To answer this question, we searched for studies in which species interactions are mapped as traits on phylogenies. Then we inferred the age of an interaction from the age of the corresponding trait on the phylogeny. For example, Peters et al. 2018 built this time-calibrated phylogeny of Hymenoptera and mapped the lifestyle of this group onto the phylogeny. Using this tree, we were able to infer that the parasitism by wasps originated at around 250 million years ago. Then we did an age comparison of clades of antagonists versus mutualists. We found that the oldest mutualisms are much older than the oldest antagonisms. This shows that some mutualisms can be extremely persistent over macroevolutionary timescales, and perhaps more persistent than any antagonisms. However, animals seem to be an exception here, in which the oldest antagonists are much older than the oldest mutualists. We think that this reverse pattern in animals may reflect an extremely early origin of antagonism in animals, instead of higher evolutionary stability of antagonisms than mutualisms. So the third question I'm trying to ask is still tentative. So I will very much welcome any comments on this part of my research, as well as the fourth question I'm going to talk a little bit later. So for the third question, I'm interested in the role of phylogenetic distance of host shifts in shifting parasites' diversification rates. So since the escape and radiate hypothesis by Ehrlich and Rabin, rapid diversification of some parasite clades has been frequently linked to significant host shifts. The idea being that switching to a different host creates an adaptive zone for rapid diversification. However, it is unclear what is the role of phylogenetic distance of host shifts in shifting diversification rates of parasites. This question is particularly interesting to me because it seems to present a paradox. A shift to a more distant host may relax the pre-existing stabilizing selection posed by the old host, and this may create an adaptive zone for rapid diversification. However, a shift to a more distant host may also mean that there will be strong directional selection posed by the new host. And if the directional selection is too strong, this may cause higher probability of extinction. So it seems unclear, at least to me, what to expect when comparing the effects on diversification between a distant host shift to one that is less distant. So to answer this question, I plan to use the hosts dataset compiled by the Natural History Museum of London. This dataset contains around 140,000 entries of host plant family information for about 22,000 Lepidopteran species. And we have collected so this has been finished with the help of my friend Yuan Yi at the University of Wisconsin Medicine. So I also plan to collect species-level time-calibrated phylogenies from the internet using Scopus. Alternatively, I can also use the Web of Science. They do the same job. Here I switched to Scopus because the university I'm at right now doesn't subscribe to the Web of Science anymore, starting this year. This search will cover all major superfamilies of Lepidoptera. So far, I have gathered about 130 studies, but I would guess most of them are still not suitable for what I want to do. But it's a good start, I think. I will also need family level phylogeny of the hosts. In this case, it will be a family level phylogeny of seed plants. So I am very interested in using this new method, this new approach proposed by Braga et al. 2020. These authors, they model the evolution of host repertoire along each lineage on a phylogeny. And in this approach, hosts used by a parasite are encoded as a character set called the rest repertoire. Each possible host species has 3 possible states. And the 3 states being a non-host, which is a species that is never used as a host, a potential host, which is the species that can be potentially used as a host, but not actually used, and an actual host, which is a species that is actually used as a host. And this approach allows the user to reconstruct the ancestral state of host repertoire for each internal node on the parasite phylogeny. And once an ancestral state has been inferred for every internal node on the parasite phylogeny, gains and losses of hosts can then be identified by comparing the state of the node or tip to that of its parent node. For each host shift, a phylogenetic distance of the host gain or loss will be calculated. For each host gain or loss, the associated shift in diversification rates will also be calculated. Shifts in diversification rates will be quantified as the ratio of the rate with the host gain or loss to the rate without. And then I can do a regression type of analysis to examine the relationship between the 2 variables. And there are some alternative approaches for what I want to do here. This is still very tentative, and I'm not sure if I'm going to do this, but the idea is that there are some phylogenetic comparative methods that allow the user to identify changes in diversification rates, both among clades and along branches on a phylogeny. And these methods could be potentially very useful for what I just proposed to look at here. And then there is the fourth question I'm trying to answer. So for this question, I'm trying to answer whether specialization predicts rates at which host species are gained and lost evolutionarily in parasites. I am interested in this because there is a recent review that identified three major sources of constraint uh, on the evolution of a broad diet in herbivorous insects. This review paper by Hardy et al. 2020, which we will talk about in next week's journal club. But it seems very unclear to me whether these constraints are stronger on more specialized parasites or less specialized parasites. Yeah, so I have two alternative hypotheses here. One predicts that negative relationship where one predicts a negative relationship where more specialized parasites will have lower rates of host gain because of stronger constraints on the evolution of a broad diet. The other predicts a positive relationship where less specialized parasites will have lower rates of host gain because they are subject to stronger constraints. However, I am not very sure what to hypothesize about the relationship between specialization and the rates of host loss. This is because there does not seem to be a good understanding of what factors constrain the evolution of a narrow diet. And I will appreciate any input on this from you. Yeah, and again, this approach of modeling host repertoire evolution is especially useful here because it allows the user to model host gain rates and host loss rates separately. And here is, and this is a graph showing what I plan to do. So first, I think I will need a time-calibrated parasite phylogeny. Then I can cut the whole tree into several subtrees of the same age. Then for each subtree, I can quantify three variables. And the three variables being host specialization, rate of host gain or loss, phylogenetic distance of host gains or loss. And after this, there should be three variables associated with each subtree, allowing statistical analysis to examine the correlation among these three variables. And at last, I wanted to talk about how a network approach can be especially useful here. I think there are many exciting possibilities. For example, one thing I have been thinking about is do different relationships between host range, breadth, and the rates at which parasites gain or lose hosts lead to the emergence of distinct network structures at the clade level? And can these network structures be used for testing fundamental hypotheses regarding host range evolution? And of course, I would love to hear what you think about this. And with that, I'd like to thank my PhD advisor, Dr. John Wins, and many other people who have been very generous with their help that made this work possible. Thank you, and please let me know if you have any questions and comments. Thank you.
Thank you so much, Yichao, for this very interesting talk. I think we have time for questions. I do have many questions to you and some ideas on network analysis that could help, but I will give the priority to people that will not necessarily interact next week with you. Okay, so questions, guys? There is some questions in YouTube. Okay, Tayo, you start, and then I will go back and forth between here and people in YouTube.
Hi, Yichao. Thank you for your talk. Very, very beautiful, your work. I have one question about your first question. So you find that positive interactions increase the diversification rate, and negative interactions decrease the diversification rates. So my question is about the speciation and extinction. So the negative interactions have less speciation and more extinction, the positive interactions. And what do you find about this?
Yeah, so yeah, I think that's a very good question. We couldn't tease apart the effects, we couldn't tease apart the process of speciation and the process of extinction. So we looked at net diversification, which is speciation minus extinction. It's a very good question worth looking into, but we don't think we could answer that question with the data we gathered.
Thank you.
Okay, Yichao, related to that, Carolina Schlossler asked a question on YouTube. That is, you mentioned that competitive interactions may have a negative effect on diversification, but competitive interactions can also fuel speciation through niche differentiation, isn't it? So what would be the biological mechanism that would explain how competition amid favoring speciation would end up leading to a negative effect on net diversification? It's just the increase of extinction. So this is my guess, sorry.
Yeah, that's so our paper is not able to, the data we gathered are not able to answer that question. So we were mostly looking at patterns, and yeah, we were being pretty ignorant of the mechanisms, to be honest. But I do have some thoughts on that. I think it's so I think most of the evidence that competition can fuel diversification is focused on intra-specific competition. But when you look at, when you have competition between clades over macroevolutionary timescales, at least in the data we found, in the data we gathered, we found that the competition tend to decrease diversification rates. Yeah, I think that competition can certainly cause extinction, and there are many examples of that already. In addition to that, competition can also slow down the rates of speciation, because when you don't have, and there's this concept of ecological opportunity. When you have a lot of competition with another clade, you don't have much opportunity to diversify, and that could slow down the speciation process. Yeah. I think that's
Yeah, I think that you point out a very important aspect of competition in your analysis, isn't it? Like it's competition among species. It's not inter-specific competition. So it's actually, as you said, an interaction that forbid the niche spread of a given species and consequently of a given clade, because we are not talking about a diversifying process in this scenario, isn't it? Like it's not a population branching out in different polymorphic through different polymorphisms due to intraspecific competition.
Okay. Can I just quickly jump in?
Yeah, and you can use that
just to make a comment this and then go back.
Yeah, but then you can make your question, Thiago.
Oh,
just
okay.
after that. Okay.
I think Yichao touched on it. First, thank you very much for the talk. It was really, really interesting. You have an amazing PhD to pursue with very different questions. It's really fascinating. I think what he mentioned also will be interesting to look into your analysis to see if the competition you're referring to when you analyze this data is between clade competition or within clade competition as well. Not only intra-specific competition, because the between clade competition is competition imposed by a species which will not affect the speciation rate of the clade you're measuring. And that's a different thing than if the competition is being imposed by a clade which will feed back and potentially affect the speciation rate which you're measuring, right? So you have two clades, you measure the speciation rate here, you measure the speciation rate here, and then you see if competition changes those rates. It'll be nice to, I don't know, I'm not sure if you have enough data points to go back and see how many of those are within clade competition and how many of those are between clade competitions, but that would be interesting, an interesting side, because between clade competition is likely to restrain or limit the diversification of the other clade. Within clade competition might act as, even if it's between species, might still act as a motor for diversification in the beginning. And another side, which would be interesting to know if you have any thoughts on this, is that we typically measure the effect as a unidirectional effect, right? This thing does this, it either increases or decreases the rate. Have you thought about ways of actually looking at more complex outcomes? I mean, it might be the competition increases the speciation rate at the beginning of a radiation, and then it decreases speciation, right? So it'd be nice to know if that's a way to go. Like, I mean, we tend to simplify things because it's hard to study, and then we pick up but it would be nice to eventually to get to the point where we can actually look at bidirectional effects or more complex effects. Anyway, I had a quick question about the first, well, I have many questions. I will just ask one, and then if I have time, I'll go back to other questions about other people. In the first part, when you look at the effect of the sign of the interaction and then the outcome of the diversification, when you say diversification rate, are you talking about an average rate or a rate at the tips? That's one question. And then the other one is, do you have any idea why? Because it seems that the Jablonski paper was a bit more uncertain about the direction of the effects, right? It had the same kind of interaction going, either increasing or decreasing the rates of diversification, while your study suggests that there is more predictable direction. Do you have any thoughts on why of those differences? Why you seem to catch a more predictable result, while Jablonski had more unpredictable or more complex outcomes?
Yeah, thank you very much for your comments and questions. So we were we tried to be as inclusive as possible when it comes to including studies. So we didn't really require the diversification rates to be a specific kind. For example, if it's a tip rate or it's a rate for the clade, we were mostly looking for rates that are associated with states of species interactions. If there's a rate that we can clearly, or at least the authors think can be clearly associated with a state of species interaction, we include that study. And yeah, so there's certainly it's a simplification, and it needs to be done very cautiously, I think. And maybe we were oversimplifying a little bit. Yeah, and that's for the first question. And for the second question, the Jablonski paper is a narrative review, I think. So it tries to connect the readers with as many papers as possible. And so I don't think it has a set of very clear criteria of including a set of clear criteria of which studies to include or which studies to exclude. And our study does just that. We had a very clear set of criteria, and what we show, I mean, the title of that paper is, we try to make it short so it doesn't actually contain all the details. I don't think it contains all the details of our major findings. I think what we actually found was that when you look at the effects of species interactions at a certain scale, and when you're very strict about what counts as evidence and what doesn't count as evidence, it seems that the effects are kind of predictable. But when you include everything, it becomes very chaotic. So we had to make many simplifications for that study. Maybe sometimes oversimplification, I would say. But yeah, that's
fair enough. I mean, it's how it goes. No criticism on simplifications. It's one idea when you're talking, maybe I'm not sure if there's enough data points, but you could go back to your analysis and see try to see if the type of method that was used to estimate the diversification as a cofactor, it does explain something about the outcomes. Do you preferentially get a positive response if you use, I don't know, BAM, or if you use Sanderson and Magalon, or if you use Clades or whatever? I mean, I'm not really sure if I do have an expectation, but it would be a nice way to sort of search to at least see if the method used explains a little bit of the results or not. Hopefully it doesn't. I mean, it doesn't matter which method is.
Yeah, that's we did that.
Cool.
So when you do that, the sample sizes get even smaller.
Small.
Yes, but we didn't notice anything.
Cool.
Yeah, it's not really, because I think the results of that paper, because we have both quantitative and qualitative results.
Mm-hmm.
So I guess the methods, I mean, the quantitative results can depend a lot on which method you used, but the qualitative results it seems pretty robust across methods. Yeah, thank you.
Very cool.
For that question.
You would have more questions, but let's circle around if there's not enough questions that we can come.
Okay. So I will ask one question. It's related to the escape and radiate hypothesis. It's something that Thiago and I and Rodrigo have been thinking about a bit in the last months. And you mentioned the possibility of using network analysis to try to explore the relationship between ecological interactions and this and macroevolutionary processes. So my question is related to one of the most fuzzy aspects of escape and radiate hypothesis, is how escaping radiated hypothesis predict phylogenetic patterns of among interacting groups. And as John Thompson always emphasizes, it do not predict like co-phylogenies or parallel cladogenesis, but it actually predicts some level of phylogenetical signal. And what I would guess would be that you find, if you describe these two clades as a network, you would find modules, and those modules would be, these groups of interacting species would show a strong phylogenetic signal. The problem, though, is that a different clade of herbivore can colonize the same groups. Two different clades of herbivores can colonize the same host clade. So, do you think that by combining your analysis to this modular structure of plant herbivore interactions, you could identify some signatures, some footprints, some fingerprints of the escape and radiate dynamics? Sorry, it was a bit convoluted, my question, so I apologize for that. I was just thinking and trying to organize at the same time. For me, it's too fuzzy, actually, the whole idea of phylogenetic patterns and in escape and radiate dynamics.
Yeah, I'm not sure, honestly. I'm just getting into network analyses. So it's very interesting. It's a very interesting question to me. But I can make some guesses, but I don't think that will be of much value, I guess.
Okay, alternatively, like using the approach that you have been using, maybe you would find a phylogenetic signal, but in a particular scale of the phylogeny. It would not be parallel cladogenesis. It will not be like the entire phylogenies as two tangled trees describe the diversification of groups, but you would, like for sections of your phylogeny, you would find some matches between clades. Is it possible with the approaches that you have been using to actually detect this kind of signal? So some association among plants and herbivores that are a non-random association between plants and herbivores of particular clades, but not a perfect match from the phylogenies or a strong signal that is spread through the entire clades analyzed?
So by using the methods I have been using?
Yeah.
So I haven't used any of the methods that I just talked about. I just thought, I mean, that's my plan. I mean, so
Okay,
sorry.
yeah. Okay, the methods that you are planning to use. Sorry about that.
Yeah. Oh, yeah. Okay. Yeah, I think there could be something to be found by using those. Yeah, especially I'm very interested in finding something very general about the evolution of host range. So I think, and of course, the escape and radiate hypothesis is arguably the most important hypothesis in this direction. So
yeah, in this sense, the analysis by Mariana Braga and others, like exploring host range macroevolution in butterflies, could be a nice system to explore how small changes to use as a reference from how small changes in how host range evolve would lead to different macroevolutionary signatures for the involving clades. Like this idea that some organisms can keep the potential of interacting with ancestral hosts, and then they oscillate in host range across macroevolutionary time. So this is a very interesting hypothesis. I think it's really hard to test though, but this might be just my prejudice related to that. I'm just my ignorance related to that, to be more accurate. Thiago, do you have any question? We have time for
I mean, we have. So I was curious about I think I missed a bit, but how do you estimate the age of the interaction? That's ancestor trait reconstruction, is that?
Yes, that's the idea. So very often you can find traits mapped on a phylogeny. Some very often people are interested in mapping the evolution of a trait on a phylogeny, and very often those traits can be associated with species interactions. For example, in wasps, the evolution of the stinger at the end of their abdomen is associated with the evolution of a parasitic lifestyle.
Sure.
Yeah, that's how we infer the age of species interactions. Of course, this method has its limits.
My question was, is it the oldest, so the age for a given interaction, let's say you have a phylogeny and you have that interaction popping out in different species at the tips, and then the age is the oldest age is the oldest, so it's ancestor state, it's the oldest age, right? Because it could have evolved multiple times independently in different lineages, but what you call the age is the oldest age you see for that phylogeny. Is that how it goes, or?
Yeah, so that's a question I ask myself sometimes. So what counts as an origin? So it's I would say we had to make some subjective choices when working on that paper, because sometimes, the changes in species interactions are gradual instead of like abrupt. So it's really hard to tell whether I mostly we just went with the oldest age for a specific type of interaction, but what qualifies as the oldest age, there's still some subjectivity there.
Sure, cool. Yeah, just I guess you're aware, right? Because it's hard to disentangle this ancestor state from the fact that this could have changed in the middle of the branch. It could have changed closer here. So it's hard to, but anyway, we have to make simplification. I was just curious about how you deal with that. Cool.
Thank you for your question.
You're welcome.
I think that the same is true for categorizing the interactions between mutualistic and antagonistic interactions, isn't it? For some antagonistic interactions, there is no question about their role as a negative effect on the prey, but for others, this is much less clear. And for mutualistic interactions, this is, as you show in the beginning of your talk, there is always this the potential for shifting the outcome of interactions, even across years or seasons. So this is, but well, that's, you need to simplify to the analysis. There is no way around that. But one thing that could be very interesting is to explore if different types if we could in some ways try to estimate this kind of turnover, like how the interactions shift in a clade, the turnover between the effects of the interactions, it would be very cool to explore. If there is some interactions that are going back and forth, and if this has any consequence to macroevolutionary patterns. I have no clue. It's just I guess.
Yeah, I know there are some studies on the evolution of mutualistic versus antagonistic symbioses in bacteria, and they found that there is repeated transitions between the two states over macroevolutionary timescales. And yeah, there's some very interesting, I mean, yeah, that could be very interesting. Yep. Yeah.
Yeah. The same is true for like some types of pollinating insects, isn't it? Like for some for fig wasps, this is quite stable in phylogeny if you are a parasite of a fig, or if you are a mutualistic fig wasp. But then the close related clade, like the Greya polytella, and then we move to Lepidoptera, they have like the eucamophs and Greya polytella. And eucamophs, this is very stable, like the mutualistic interactions are very stable. But when Greya, there is a lot of change through time. If I remember correctly, the eucamophs are much more stable in their patterns of interaction. So maybe you could use like this replicated evolution of the same interaction, like this kind of parasite form of pollination that some insects evolved, to actually explore the role of mutualistic and parasitic interaction in diversification in a very general way. Because this type of interaction evolved multiple times, isn't it? Like they have in fig wasps, you have in Lepidoptera a couple of times, and other groups of insects do the same. So,
yeah, thank you.
Yeah, you're very welcome. So, Yichao, if we have no further questions, I think that we actually passed our time already. Yichao, thank you so much for your talk. We catch up later about some ideas. I need to think more carefully of some points you mentioned in your talk. And thank you again. E muito obrigado a todo mundo que veio e assistiu a palestra do Yichao. Foi prazer. So that's it. Thank you, guys.